Good morning to those joining us from North America, and good afternoon to those joining us from Europe. Welcome to this webinar, Primary Sclerosing Cholangitis, Research and Predictions. This is the 7th and final webinar in the PSC Partners 2020 Annual Conference Webinar Series. My name is Mary Diaz. I'm the president of PSC Partners Seeking a Cure Canada, a Canadian registered charity and affiliate of US-based PSC Partners Seeking a Cure. Thank you again for joining us. Thank you to our expert panelists from Colorado, Canada, and Norway. I'd like to now pass the Zoom microphone to CEO and founder of PSC Partners Seeking a Cure, Ricky Safer.
Thank you, Mary. Thank you very much to everyone for taking time out of your day to join us for the last webinar in the PSC Partners 2020 conference series. We know the timing is inconvenient for some people, but it's what we had to do to assemble this amazing group of international PSC experts today. As with all our past webinars, this session will be recorded and posted on our website. Additionally, this is definitely a period of confusion for most of us. Between the COVID-19 pandemic, the uncertainty of the US election, and more, 2020 continues to be a time of uncertainty, fear, and unrest. We appreciate that the expert panel and our patient community are dedicating an hour and a half today to discuss PSC research and predictions. PSC is not taking a break, and neither are we. We've all been disappointed in having to shift this year's 2 in-person patient caregiver events to virtual events, but we're hoping that in 2021, we'll be able to again travel safely and get together in person. Today, I'd like to thank our conference co-hosts, University of Colorado Health and Children's Hospital Colorado, as well as our other corporate sponsors, Gilead, Mirum, Intercept, Alpareo, Shire, and High Tide for their support of our virtual conference series. We are thrilled to present our 3 expert international speakers, Dr. Johannes Hove of the Norwegian PSC Research Center, speaking about his research on the gut microbiota in PSC, an update. Dr. Gideon Hirschfield of the University of Toronto, presenting on risk stratification tools, what does it mean for me? And Dr. Tom Carlson, also of the Norwegian PSC Research Center, discussing drug development in PSC, where do we stand and where do we go? Now, I'd like to introduce our moderator, Dr. Lisa Forman, Professor of Medicine, Gastroenterology, Hepatology Program Director at the University of Colorado, Anschutz Medical Campus. Dr. Forman heads the Autoimmune Liver Research Program and has been the primary investigator of many clinical and epidemiological trials and studies. I'll turn it over to you now.
Thank you, Ricky. I'm really, really honored to be part of this wonderful group that you have developed, and I'm really happy to be able to moderate today's last session. As we all know, it's been a very interesting year, and we were really disappointed not to be able to host the annual PSC conference, but again, with Zoom, it really allows us to invite these international speakers today when we otherwise would not have been able to do. And so I'm really excited for today's panel of experts. It's really quite a powerhouse, and of all the webinars that we've had, to me, the future of PSC and research is really, in some ways, the most exciting and the most hopeful. We're really kind of in a new era, I believe, for PSC research, and so I'm really looking forward to hearing our 3 speakers today. And again, as Ricky said, it's Dr. Hove, Dr. Karlsen, and Dr. Hirschfield. So with that introduction, I would like to introduce our first speaker, which is Dr. Johannes Hove. He is a researcher at the Norwegian PSC Research Center. He received his MD from the University of Oslo in 2003 and his PhD in 2011. And he is leading the research group Genomics and Metagenomics in Inflammatory diseases, and his main research focus is on the role of gut microbiota in PSC and other inflammatory conditions. And so that leads us to his talk, which I'm sure will be very exciting, on the gut microbiota in PSC. Dr. Hove.
Okay, thank you very much, Lisa and Ricky and everyone, and thank you all for joining this webinar. And I'm extremely happy to talk about my favorite topic, the gut microbiota in PSC, and I'll try to provide an update. And even for some of you that may not even have the basics, I'll start with a brief introduction. Next slide. Next slide, please. So the plan for the talk is, of course, if you want to update on something, you really know, we need to know where we are. So I have to start with that, and also to explain to you kind of the aims of the research that we do. And then I'll try to highlight, I would say, some recent scientific observations of key interest from the last couple of years, and how these may help us accomplish maybe something that could even be clinically important, because that's of course the main aim, to be able to understand something that may actually influence the everyday life of patients with primary sclerosing cholangitis. So I'll touch upon something called Klebsiella and the gut leakage, and we wonder a lot about what the bacteria are actually doing. Are there fungi in the gut? Are microbes in bile important? And most importantly, of course, can we treat this? And I'll give you a take-home message. Next slide, please. So this is the microbiome. You may have read about it, maybe not, but the microbiome is really this major organ of microbes. I focus most on the bacteria, but there are also lots of viruses and fungi. And there are lots of them in the gut. The majority are in the colon, but you also have lots in the oral cavity, as you can see, and all along, actually, the intestine, you will find bugs. And there are these more or less equal the number of human cells in the body, and there are maybe 10 million known bacterial genes. So this is a huge amount of different functions that the bacteria can have, and just in comparison with the poor number of 20,000 genes in the human body. Next slide. So, and this is really the background from in PSC. We really tend to focus on the liver, on the upper part of the figure here, where the disease occurs. However, as you know, is that this is really a disease which is tightly linked to inflammatory bowel disease. At least in our material in Norway, up to 80% of patients have a diagnosis of inflammatory bowel disease at the same time as they have a liver disease. So this really means that this is not just a disease of the liver, this is really a disease of the gut-liver axis. And this is at least a proof that something is going on. So I guess that's one of the knowns there. Something's going on in the gut-liver axis. Next slide. So what we do know about the gut microbiota in PSC is that the composition of the microbes, at least when it comes to bacteria, is different in patients with PSC compared with healthy people. That is, individuals with no disease conditions. So this is just a figure here where we have looked at the composition of the microbiota, of all the bacteria in stool samples of patients with PSC, ulcerative colitis, and in healthy controls. And this is a measure of diversity, really. So we can see that the patients have reduced diversity. That means, for simplicity, less different bugs. And in addition, there are some specific bugs that seem to be different. In particular, we were very interested in a bacterium called Veillonella, which was more prevalent in our patients. But what this means, we really do not know. Next slide. So we know that the microbiota in PSC patients is different from healthy individuals. But what does this mean? Does this mean that the microbiome or changes in it cause disease? Or is it actually the disease that changes the microbiome? And so this is really a big old chicken and the egg problem, which is quite common. And we can't really solve it with a typical study of comparing patients and controls. And another important question is not just whether it's the cause, but whether the microbiome can actually modify disease. And this is perhaps more clinically important, because if the microbiota composition or the microbiota functions can modify how the disease in the liver progresses, then this is a potential treatment target. Next slide. And we have some evidence of at least the principle that this could happen. And this is a figure on mouse models of disease. There are many mouse models of cholangitis. They don't resemble necessarily PSC, but they show the principle of inflammation in the bile ducts. And to the left here, we can see that we have different mouse strains with and without cholangitis, and they have different microbiota composition. But then we turned these mice to the right, and we made them germ-free. And then they had less severe biliary disease. So without the bugs, when they were sterile, they had less cholangitis, which is interesting. So bacteria in the gut somehow may influence cholangitis. But there are also other mouse models where when you remove all bugs, the disease is worse. So the microbiome may obviously influence cholangitis in different ways depending on which disease mechanisms are operating. So at least the principle is here that we can perhaps modify liver disease by changing microbiome. Next slide. So where do we go? Of course, as I already alluded to, we really want to understand disease causes and mechanisms. So that's one important reason why we study this. But for clinical applications, it's really important to keep that in mind all the time, because if we're ever going to get there, we need to have that as a goal. So could it be possible that looking in the gut, the microbiota composition or other markers of microbiome, could that be helpful in diagnosing disease or monitoring disease or selecting who should have which kind of treatment? Or could we even change it, target the microbiome to prevent disease or to treat disease using diet or drugs or things like this? So these are really the aims of our research. Next slide. So then it's time for the update. What is really new here? Next slide. So the first thing I'd like to mention is what I call the Klebsiella story. The Klebsiella bacteria are gram-negative rods. You can see them in the figure on top there. And this is a bit of a complex paper that was published a couple of years ago, suggesting that gut pathobionts, that means bacteria in the gut that actually somehow often give disease, may perhaps change the gut barrier, activate immune cells in the liver, and perhaps influence the disease called PSC. Next slide. I'll just mention a few things from this. And here is a figure on one of the experiments they did. Here they have mice that in principle were germ-free, 3 different groups. And each group got a different treatment. So one group didn't get any microbiome, only kept germ-free. One group got their new microbiomes from healthy people. So got them humanized with healthy microbiomes. And then you had one group of mice that received microbiomes from patients with PSC and ulcerative colitis. And then after some time, they all got a diet with DDC, which is destructive, damaging to the bile duct, so they got cholangitis. And the end point of this experiment was really that those who got those mice who had cholangitis model, but with them microbiomes from PSC patients, they had more severe biliary disease and had more markers of gut leakage like endotoxin in their bloods. So it seems like the microbiome of PSC patients with ulcerative colitis versus this model of biliary disease. Next slide. And then they went on to dig into the gut, to the abdomens of these mice, and they were able to grow some bacteria in these mice that got microbiomes from PSC patients. And one of them was a Klebsiella species. And then they went to human patients with PSC and found that Klebsiella apparently was more prevalent in the fecal samples of PSC patients. And then they did some experimental studies showing that these Klebsiella bacteria, at least some of them were able to make small holes in the lining the cells lining the gut, perhaps explaining the gut leakage, and perhaps then explaining the more severe biliary disease. Next slide. So to summarize here, this is of course, you have the gut-liver axis to the left, and then to the lower right, you really have a figure of the gut barrier. And for some reason here, then these Klebsiella pneumonia make holes in this lining, so you get gut leakage. And this, as you can see to the left, all the blood from the gut enters the portal vein and then enters the liver. And in the upper right corner here, you can see then the figure of these Klebsiella and other inflammatory particles, are probably then driving inflammation and immune cell activation, etc., in the liver in this model. So specific bacteria induce leakage and may lead to immune activation and worsen bile duct disease. Next slide, please. So there are some questions after this study, I would say, because they don't really show that these Klebsiella make induce gut leakage also in humans. But that would be important to find out. And how common are these bacteria in our patients? Is this really a common mechanism? But it's still a very promising concept, but because it could be so that at least some patients with PSC have specific bacteria in their guts that influence disease course. And if they do, then we could perhaps target treatment in this subgroup and destroy these bacteria using antibiotics and other treatment methodology. Next slide. So my next point would be about gut leakage, which is also then more or less related to the previous observation. Considering this strong link with the colitis and PSC, many people have been talking about gut leakage as a phenomenon in PSC, and then bacterial products enter the liver and induce inflammation. And we see then the principles in the previous study. However, in human PSC, there's actually very limited evidence of this going on, but we do not have very good methods of measuring it. We did one study, which we have figures from on the top to the right here, where we measured some markers in blood that could perhaps reflect increased gut leakage. There's something called soluble CD14 and LPS binding protein, which were both elevated in PSC. And as you can see in the lower panels there are some curves showing that those with high levels of these markers, they had shorter time until they needed a liver transplantation or deceased, unfortunately, from their condition. So, so this is possible here that these markers and this phenomenon actually represent gut leakage, which could then be a mark of severity in PSC. However, they're not very specific. Could also be more inflammation, or just representing inflammatory phenomena. And it's also that other gut leakage markers were not elevated. So I think we need to study this further in our patients. Next slide, please. I think one important question that we have been asking ourselves many times is, what are the bacteria actually doing? And I try to usually show this figure to explain some of this. This is really a major biochemical factory with 100,000 billion bugs. And then everything you eat and all drugs, everything really meet the bacteria and other microbes first. And they have a massive metabolism. They digest, they help us digest and utilize the energy of the food, but it also generates new products and lots of byproducts. And many of these may, as shown here, go from the lumen and to the bloodstream to the liver and actually also to the rest of the body. So it's highly likely. There are many examples now of byproducts of bacterial metabolism of dietary substances actually influence our different organs. So this is a potential mechanism by how altered gut microbial functions could influence the liver disease. Next slide. So we have studied this by studying all the bacterial genes in the gut. Not only which bacteria are there, but actually which genes they carry. And here you can see a figure actually showing that there are less different bacterial genes in the PSC guts. So the bacteria in total have less functions. It's sort of a diversity measure as well. And as you can see here on the figure, this study was really, this was supported by PSC partners, so we are very happy and thankful for this. Next slide. But I think one of the main observations we have, this is one example to the left, is of one gene. That's one function. This function, this gene, actually is one of those that helps the bacteria produce some vitamin. This is the example, vitamin B6. And we also observed many other of these metabolism related to essential nutrients. That means nutrients that we actually need to eat or we need our bugs to produce, like vitamins, some amino acids, etc. And these were, many of them were reduced or changed in PSC patients. And we ask ourselves whether this is relevant for disease, and we're really studying it hard and hope to publish on this very soon. Next slide. I think another interesting observation recently is the question of whether we also have fungi in the gut, because it's not just if we have, because we definitely have. There are fungi also in our guts. The figure to the left is supposed to show differences in size. So fungi are a lot larger, perhaps 10 times larger than the average bug, bacterium, but there are a lot fewer then. So there are a lot fewer to study. And there was one, as you can see to the right here, there was been one good study on the fungi in the gut of PSC patients, and there are some examples of fungi that were different in PSC patients compared with healthy. And obviously there are some. What this means, I'm not sure we can say. But these may interact with the bacterium and in total perhaps contribute to the dysbiosis or changed environment in the gut of PSC. Next slide. Okay, and then one question that is not directly in the intestine, but about bile is whether microbes in bile are important. They obviously are for many PSC patients, in particular if you have really big trouble with cholestasis, where you have strictures and bile accumulating in the liver. You may have infections because you have problems with draining the bile into the intestine. So this is, of course, one problem with infections, but could it be also that the bile, also when they're not infected, could have bacteria that somehow are driving disease? And this is maybe difficult to study because there are a lot fewer bacteria in the bile than in the stool, so it's prone to contamination, etc. But there was one good study on this from Germany, published this year, where they looked at the microbes in the oral cavity and in the duodenum, just outside the bile duct and in the bile duct. And they really see that the bile microbiome was specific for bile. And it was changed in PSC patients compared with controls. And you could see that it was somehow associated with the composition of the bile. What this means, I would say, is also a bit unclear. Is this just colonization because you have these changes in the physiology of the bile duct? Is it because they have been infected? Or is this actually driving disease? I think this is an important area of research. Next slide. So this is the final topic here. Can we treat this? We still really don't know what would be important to treat, I would say. And that's perhaps the main caveat when we talk about whether we can treat it. But still there have been some attempts. You probably heard about fecal microbiota transplantation because it's such a spectacular phenomenon, where you actually give patients microbiota transplantations from stool from healthy individuals, either using a oral duodenal route or as using a colonoscopy. So you can do it both ways. And there was a landmark pilot study from Boston, from Allegretti last year, where they had 10 patients who received FMT using colonoscopy. I don't think we can really use the numbers to say anything about whether this affected their liver disease or their liver biochemistry, but at least you could see, this is the figure on the bottom here, that it increased the diversity of their microbiota. So it definitely changes the microbiota, and then you would need a lot larger study designed perhaps differently to say if this is of help at all. But you can perhaps use this type of pilot studies to understand something about disease mechanisms and whether this actually has an influence on liver disease. Next slide. So the other question, which is of great interest, is whether we can treat it with vancomycin. There's been a lot of talk about vancomycin. Which is an antibiotic that kills largely gram-positive bacteria, so it leads to really major change in the gut microbiota. There have been 2 short-term trials where you can see that it influences the alkaline phosphatase, that's liver biochemistry, short-term. But we know very little or nothing, I would say, on whether this actually influences the liver disease. So this is the type of evidence that we would really need before this type of treatment could really be applied generally, because vancomycin as an antibiotic, many would be skeptic to prescribing this without really good evidence that it works on the liver disease, because of the potential side effects or potential resistance development and potential change of the microbiome that we really don't have the overview of. But this, I think, it has a very strong rationale to study this. There is a study ongoing, so I hope this really can be completed in a good manner, so we will know more about this. To the right, you show a figure here, because vancomycin not only kills bacteria, but there is also some evidence that it has direct effect on immune cells. Next slide. And I'm actually at the end of this update, and I have tried to present some, I would say, key observations the last year, which then adds to what we already knew, that the gut microbiota are different in PSC. But I think based on these observations, we can ask ourselves, maybe in some PSC patients, the presence of particular bacteria, like Klebsiella, may actually be driving disease. We don't know this for sure, but there is a potential. This could be true. And if so, it has important implications. And then it's the question that the gut microbiome PSC really has altered metabolism of multiple essential nutrients. Is this important for disease? I have a feeling it does, but it also remains to be shown. And finally, I think definitely there's a potential for gut-targeted treatment for PSC, as I mentioned here at the end. But in my opinion, not yet. We need to know more first. So, but I'm a cautious optimist that this will be relevant in the end. Next slide. So this is real acknowledgement. This is the group, the people doing the work. I'd like to thank also the funders, and then in particular PSC partners, for funding our very important metagenome study that I referred to here. Thank you very much.
Thank you. Thank you for doing this type of research. We will look forward to how it all turns out. So the next speaker is Dr. Gideon Hirschfield. He's always a wonderful speaker. He's a clinician scientist based at the Toronto Centre for Liver Disease at the University of Toronto. And he really has a dedicated autoimmune liver disease practice. And his interest is looking at new therapies for patients living with autoimmune liver diseases. And today he's going to be talking about risk stratification tools.
Well, thank you, Lisa, and thank you for all the organization that goes behind today's webinar. So it's a pleasure for me to put my clinic on hold for an hour to give you a talk on PSC, and it's a great pleasure to be able to give that talk with two of my colleagues and friends from Norway, and really for me to learn, because even just listening to the first talk, I thought of some questions about what we should do for patients living with PSC. But my topic is really about risk stratification, and just to give you some insights into how I think about risk and why we think about risk in PSC, and why as a patient one needs to have some concepts as to understand where you are on your journey. Next slide. So of course, living with PSC is like living in a vortex, and it's a vortex where there are lots of different aspects, where the patient's in the center, and the clinician and the patient support networks and the carers are thinking about monitoring. They're thinking about how to explain the disease. They're thinking about whether the disease is being managed to the highest quality. And as you heard, we're thinking about research because we want new treatments. And this means living with a number of aspects. And one aspect in particular is fear and uncertainty. And managing fear and uncertainty can be made easier by understanding your own individual risk and where and when things may happen to you. Next slide. And the imperative, of course, is, as you know, we don't have specific treatments for PSC. We do a lot for our patients around imaging and surveillance. We have a very highly successful program in transplantation around the world for patients with PSC. and our patients benefit from antibiotics and they benefit at the right time from ERCP. But the therapies that we really want are the therapies that are disease targeted, that are anti-cholestatic, anti-inflammatory, and anti-fibrotic. And urso is a drug that we thought might help, but many of us feel it doesn't. And so we're looking for new generation therapies. We're looking for new clinical trials, and we want to put the right people into those clinical trials. So there's an ongoing need for rational, targeted, next generation therapy that is more efficacious, that is well tolerated, and more to the point that we help our patients choose to take part in the trials at the right time for their disease, not just so that they individually benefit, but so the community of patients with PSC get the most from clinical trials. Next slide. And the challenge is that our patients live with ulcerative cholangitis. The slide's actually pinched from Tom Carlson's review in Nature Reviews Gastroenterology and Hepatology. And increasingly, many of our patients are having spyglass cholangioscopy, and we're actually seeing the disease that we are challenged by. And we're always struck, I'm always struck when I see this picture, how the bile duct looks similar to the colon. This is a disease of the bile duct and often the colon, where there is cholangitis and colitis. Next slide. And as we pick away at this disease, we like to think about the 6 C's of PSC. It's covert. So we're picking it up often late in the biology, but early in the presentation. You may not have symptoms, and the abnormalities may only be seen on MRI and your blood tests are normal, or it may be all the way down the line where you're already yellow. You've got inflammation in your bile ducts, and that inflammation is driving the disease, but is also secondarily impacted by infectious cholangitis from the bile getting secondarily infected with bugs. And there's many causes that we think about, which you've already heard about the microbiota. When we don't treat this very well, and we don't treat it as well as we'd like to, our patients end up with cirrhosis. And along this journey, they're often living with inflammation in their colon, and their colitis needs to be managed at the same time. And sadly, one of the C's that we don't like of PSC is the cancer risk. It's real, it's impactful, albeit the majority of patients don't get cancer. It nevertheless is a major cause for uncertainty, anxiety, frustration, and worry. And yes, everything is cure. We can thank Ricky, and we can also thank the UK PSC charity, because they told me that they wanted, when we did this little infographic, they wanted the last C to be cure, because that is what we're after. And as you'll hear from Tom, there are a number of approaches that we're focusing on. Next slide. And in the cycle that you live in, there is immunity and aberrant inflammation, there is cholestasis, and there is liver fibrosis. And all of this is a disease which loosely is one of immunobiology, the interactions between the immune system and the bile system. Can you hit the slide once? That's the liver enzyme ALT, AST that we can measure for you, that tells you how much inflammation there is. Can you hit it again? The ALT, phos, and bilirubin tell you how much biliary damage there is. Can you hit it again? And your spleen size and a falling platelet count tell us how much liver fibrosis there is. But when we try and predict your risk and where you're going, we look at these blood tests. But in addition, in PSC, there are 2 other factors that we make it much harder to be accurate. Next button. That's your cancer risk, which actually unfortunately appears to be independent of your disease progression risk. And next button. Then it's also your colitis activity, because it's quite clear, although hard to prove in prospective studies, that when there's active colitis, there can be active cholangitis. Next slide. So where do I stand? This is the MRCP of a patient with PSC. You can see that the biliary tree doesn't look healthy. They're already on biologics for their colitis. They're not jaundiced, but their ALT phos is elevated. The ALT is slightly up. Their FibroScan is not normal, but it's not abnormal. Next slide. So just as the conversation has gone already this morning for my patients in clinic, and just as it's going to continue immediately I finish this talk, is this bad disease? What should I do? What about cancer? Surely URSA will help. Next slide. What does help is our collaborative efforts. And working together through the International PSC Study Group, we're able to quantify some of the risks that our patients live with. This can be unpalatable sometimes, and the challenge that today's talk gives me is to explain to you that you may not all be on the same journey. It's true that 10 to 15 years after diagnosis with PSC, nearly 50% of our patients will have needed a liver transplant. What does that mean for me? It means that I started my practice in Toronto in 2007, and I interrupted it for a little bit of time in the UK, and I come back, and many of my patients have returned to me. That means the patients that I looked after in 2007 and 2008 are now the patients I look after as they have a liver transplant. You can see the rate of liver cancers, bile duct cancers, is under 1 in 5 patients getting it, but it's still too high. And you can, similarly, that's the same for cholangiocarcinoma. Next slide. But when we dig a bit deeper, we see that there are different phenotypes. By phenotype, I mean different pictures of PSC. So everyone is heterogeneous, everyone is different. If the disease is mainly restricted to the small ducts, you do much better. If you have a Crohn's phenotype of inflammatory bowel disease, when we put together thousands of patients, the patients whose inflammatory bowel disease looks more like Crohn's actually have a better outcome from their liver disease. Same thing can be seen for some with cancers. Next slide. And the risk of bile duct cancer, that biggest fear that we all live with, and you live with even more than the clinicians caring for you, is loaded. It's front loaded. When you actually look at when do patients with PSC develop their bile duct cancers, that group of patients, we think it's around 15% in total, so 85% don't get bile duct cancer. But the bile duct cancers are actually front loaded to the early part of their disease. And statistically, although not universally, the bile duct cancers is also age dependent. It's more common as you get older. Next slide. And so we looked at this in the UK with Dr. Trivedi from Birmingham, when we tried to look at across many thousands of patients with PSC across the whole country. And we can see lots of interesting themes that come out from that data. One is that most patients have developed their colitis by the time they've diagnosed with PSC, but not all patients. And we have patients who present with their colitis more than 10 years after their diagnosis of PSC. Teaches us we've got a lot to learn. Next slide. And we can see some of the features again, which we have amplified from the original International PSC Study Group now to this paper that we published on UK, where really youth demonstrates one of the biggest risks for outcomes of PSC. Our young patients are having a disproportionate burden. There's a heritage difference. In the UK, Afro-Caribbean patients were doing worse. There's a gender effect. Female patients with PSC do better. And pleasingly, although we don't really know what this means precisely, our patients who get more scans seem to have less chance of dying from cancer. So our surveillance strategies do seem to help our patients independent of their risk stratification tools that we might use. Next slide. And really the thing that came out of this paper was the emphasis on age. No one is ageist in the PSC community, but this is a disease that disproportionately impacts younger people. So we see patients with PSC across the whole age. I think the oldest patient I'm seeing in clinics today is nearly 80 with PSC. The youngest patient will be 18 because I'm an adult physician. So I look after between 18 to 80. But when we look at the events, when we look at what happens to our patients, the truth is that if something bad happens to a patient with PSC, when they are under the age of 40, it's likely to be because of their PSC. When they are over the age of 60, it's likely to be something else that is actually leading to their illness and not actually their PSC. So a really disproportionate burden is placed on our patient, our community of patients who are under the age of 40. Next slide. So to give some concept about stratification, we've got to think about the things that we'd like to measure. We can think about things that may be modifiable. We can think about things that relate to liver tests. We can think about things that look at your bile ducts, although this has not actually been as fulfilling as we'd like. And we can actually look a large part at liver fibrosis and liver function, because actually those are the ones that really have helped us the most. And that means there are some things you can't change. You can't change your biological sex. how that impacts the biology of your bile ducts. You can't change your IBD, okay? Your liver enzymes are helpful to measure your risk. Your MRI actually is less helpful than you think, as I'll show you, but your markers of scarring are very helpful in predicting whether, as you approach the likelihood of needing liver transplantation. Next slide. And simply, alkaline phosphatase is a very simple one-off test of risk. And if you just do a very simple study, this is a study from Oxford, which didn't even have a validation group, but it showed that if your ALK phos was, more than 1.5 times the upper limit normal, then, and I think there's an error in the original paper in terms of the less than and greater than sign, but essentially the patients whose ALK phos stays high did worse. Next slide. And we confirmed this when we looked at a UK PSC cohort where we had many thousands of patients from across the UK. And if your ALK phos was, in this case, above twice the upper limit normal, outcomes were worse. But the exact threshold where we should cut the ALK phos is difficult. Next slide. And the problem is that it doesn't correlate with the treatments that we give. So ALK phos as a one-time measure does help give you your risk. But if we drop your ALK phos by giving you urso, and hope that it was the urso that dropped your ALK phos and therefore you do better, that's not what we see. And this was a big Scandinavian study. So it didn't matter why your ALK phos went down. The patients with the lower ALK phos did better. And I think it's really important for me just to make a slight detour here to explain to you why there's a disconnect between alkaline phosphatase and treatment response. So alkaline phosphatase does predict what will happen, but the change in ALK phos does not. And that's because the disease is at the bottom of your bile ducts. The tightness is at your common bile ducts. But the ALK phos response is actually, why the ALK phos is elevated, is actually probably related to an increase in pressure up all the different bile ducts, all the way into the periphery of the liver. And we can mop up some of those ALK phos changes in the periphery with drugs like urso, but we've done nothing to touch that tight problem at the bottom of your common bile duct. So when we drop the ALK phos, that doesn't mean we're necessarily helping you. And that's one of our challenges. Next slide. And this work from Palak Trivedi again shows you the problem with ALK phos. Helpful as a one-off, but it varies a lot because it varies with cholangitis. And that means it can go up and down. And this is what happened in a clinical trial. Next slide. And the change in ALK phos, unfortunately, did not associate with immediate benefit for the patients in this clinical trial over the period of 2 or 3 years. So it helped us stratify who was likely to run into problems, but the improvement or change up or down did not help us. That's not to say that if we had a great drug that normalized ALK phos for 10 years, that wouldn't be a good drug, but it's to say that it's not as easy as a drug that drops your ALK phos is a drug that will be a winner. Next slide. What about fibrosis tests? This is, I think, work that's come out from Scandinavia, from Norway. ALF is a test that's available in Europe, the enhanced liver fibrosis test. It will be available in North America soon. It's taken some time to get a license in the US for reasons which are a bit unclear. But essentially, the single blood test is a good way of measuring liver fibrosis. And the higher your ALF score, the more likely you are to run into problems. So this is a non-invasive way of measuring scarring. So it will become useful in the future for measuring risk. Next slide. And actually, in that clinical trial I showed you that Dr. Trivedi analyzed for ALK phos, we actually saw that there was some association not only with ALF at baseline in predicting risk, but the change in ALF over that time of that clinical trial did seem to associate with events. So new clinical trials are actually starting to measure ALF over time in the clinical trial to see whether your risk has changed from the new drugs. Next slide. Liver biopsy. This is a very good way of predicting risk. However, it's invasive. Our patients don't like it, but I'm afraid it has a great utility both in the development of safe drugs, looking for liver toxicity, and effective drugs. And a group of work led from Amsterdam has shown that the more fibrosis that we see on the liver biopsy, the more likely you are to run into problems. But that's why I showed you the ELF test first, because we'd like to fast forward ultimately beyond liver biopsy and move towards using a blood test instead of a needle. Next slide. What is challenging for us is actually when you've got very little fibrosis, that's very good. So I'm afraid, you've all learned what exponential means from COVID, and how you start off with just a few cases of COVID and everyone is lulled into a false sense of security, but then you turn the corner and you go exponentially. Well, in PSC and liver disease generally, as you get more fibrosis, then things start to heat up a bit. Why is this important? Why am I showing you that the idea that your baseline level of scarring determines how much disease will progress? Because it means that in the clinical trials that we're running at the moment, we may choose not to include patients with no liver fibrosis at the moment because their disease is too early and their risk of an event is not high enough and they won't show any benefit from a drug. And we may say, to get the best drug as quickly as possible, let's focus in on just a smaller group of PSC patients at a different stage of their journey of their disease. Next slide. And instead of measuring it by liver biopsy or by ELF, the other way of measuring liver stiffness, not necessarily just liver fibrosis, liver stiffness is the FibroScan. It is more available in North America slowly, but again, Europe's been ahead by about 10 years. And this is data from Olivier Chazelier in Paris, who again has collected data from across the world, showing that the higher your FibroScan, it does predict that your risk is going to increase. And that's why FibroScan is now part of risk assessment in clinic, and we do it every 1 to 2 years in my clinic, and also as part of clinical trials to get the right patients into trials. Next slide. MRCP, I'm afraid, has not been very helpful. Your biliary imaging has not given us the answer yet. I think it's going to be hard for it to be helpful. The truth is that when this study was done, and this is a study of using MRI, the thing that was most prognostic was using the MRI to pick up cirrhosis and splenomegaly. So not really related to PSC. So I'd say watch the space for MRI. It's not quite where we want it to be. Next slide. So this comes to models. What do you do in clinic? What do patients do at home? There have been models of what happens to PSC for decades. There's always a new model being published, but in fact, they're using the same information. We're looking at age. We're looking at your liver biochemistry. We're looking at how long you've had PSC and what is the nature of your PSC. And we're using that to try and give you a dial-up version of your risk and to repeat that over time so you understand whether your train is going slow, whether your train is speeding up, or whether your train really has hit full speed. Next slide. And you can do the Amsterdam-Oxford score, which is on the internet. Again, that uses very simple clinical available tests and gives you some idea of transplant-free survival on the right with a graph. Next slide. UK PSC, our score is very similar and will give you risk. And actually, in many ways, some of these scores are best for reassuring you that, in fact, you're at very low risk and and therefore you can wait. Next slide. Or the PRESSA score from the Mayo, which is using forms of machine learning and artificial intelligence. So you can't see the equation because it's really done by computer, but you can also get a graph of your risk of liver decompensation, ascites, varices, and encephalopathy. All of these things help frame the conversation for you and your clinician, help you understand where you are in your journey, and help you decide the timing of clinical trial involvement. Next slide. But to put it all together, you can see that, in fact, this disease is very heterogeneous. And this disease, therefore, actually remains to be one where we have to individualize care. And so, although I do use risk models in clinic if patients ask me, I try very hard to treat the whole patient, their liver, their bowel, and the rest of life that's going on, because that's really the most informative way to manage chronic disease with uncertainty. Thank you.
Thank you. Unfortunately, Dr. Hirschfield has to go back to start seeing his PSC patients in clinic. And so if any in the audience have any specific questions geared toward him, maybe you can send them to Ricky and he can answer them at another time. But thank you for that talk. The last speaker, I'm pleased to introduce, is Dr. Tom Carlson, also from Oslo, Norway. It's good to see you again virtually. Sorry you can't come to Denver. But Dr. Karlsen, for everyone to know, he is the research head of the Division of Surgery, Inflammatory Medicine, and Transplantation, and is also the leader of the Norwegian PSC Research Center at the Department of Transplantation and Research Institute of Internal Medicine. And today he's going to be talking to us about drug development in PSC. Where do we currently stand and where do we go? So thanks for joining us.
Thank you, Lisa, and thanks to Ricky and all of the PSC Partners team. I surely miss being at the physical meeting, talking to you face to face, but this is the way it is, and I hope that we will be able to share some reflections during my presentation here, even though some of you may be distracted by ongoings regarding elections in the US right now. It's a kind of a summary talk, and I will be focusing also forward looking as Johannes and also Gideon. So next slide. I just wanted to start with an inspiration because, as some of you or many of you will have recognized, the Nobel Prize in Medicine and Physiology this year was awarded for the discovery of the hepatitis C virus. And I think this is an awarding which was not only done because of the identification of the virus, which of course was an interesting observation, but also as much, I would say, to the journey that followed the identification of the virus, where you since the late 1980s, early 1990s, until 4 or 5 years ago, you saw a massive research effort throughout the entire liver community, ending with drugs today, which actually cure the virus, get rid of the virus. It's in medicine. It's quite unprecedented journey. You come from a disease where you actually don't know what's going on. You find a cause, and you delete the cause, and you cure the patients. So I think we should keep this as an inspiration, and we will revert to the slide at the end and see how we're reflecting upon this in the context of PSC. So next slide. As Gideon ended his talk saying, there is a lot to accomplish here. It's far more to be said about achievements in PSC than in the eradication of a virus. In many ways, the hepatitis C research have had, I would say since the discovery at least, a pretty simple task. What they had to do was to get rid of the virus. In PSC, we have to do so many things, and we have to do them all at the same time. We need to improve all these parameters that Gideon was referring to, ALP, various immunological markers. We need to improve, ideally, what's going on in the tissue, the histological situation, both in the bile ducts, but also in the liver, surrounding the bile ducts. We need to make sure ultimately that patients don't go on the journey that Gideon was referring to into a future liver transplant, and we have to get rid of cumbersome symptoms during the treatment period. That includes IBD, which was clear from Gideon's talk, but it also includes a variety of other symptoms: itching, being tired, and the overall situation in the bigger scene of life. And as the last 2 points here say, not only do we have to give a weekly cure or a 1 week's cure or a 2 months course of prescription, this is a chronic disease as far as we are with our understanding today. And the treatment has to go on for years and years and ultimately decades in many patients. And of course, for such therapies, you have to pay particular attention to side effects. If you have something that you prescribe for a week, you can tolerate some side effects and then therapy is over. But when you have to take the drug every day, year in and year out, the side effect story is becoming an important one. Next slide. And this is in many ways where the whole journey of drug development in PSC started. And I will not go to the beginning, but I will show these two figures. And these are survival curves, which we have now seen many of those from Gideon. And the upper left figure shows the results of this big Scandinavian ursodeoxycholic acid trial. It's the biggest trial performed. It was still not big enough, probably. And in the end, even though there seems to be a small trend, if you look at the y-axis, it starts at 0.8 and goes up to 1.0. And this very, very small difference that you see there, it was not significant. So in many ways, we don't really have definite evidence that urso impacts on transplant and survival in patients with PSC. Then there was this appearance of the Keith Linder trial in 2009, where you actually saw the opposite effect. And what was done in that trial was to increase the dose of urso, because if something is good, then maybe giving even more of the good thing is even better. But actually here, it seemed that patients with the higher doses, they had more complications and more problems than the patients not receiving any urso at all. So if you go to the next slide. So I think that what we can conclude so far is that certainly the high doses of urso, we should avoid them. But this trial from Keith Linder led to a big debate, which I would say is still ongoing, on how do you then use urso in the PSC patients. And you can see from the North American development that many patients were on urso prior to the Linder trial, but more than half of the patients were taken off urso, not given urso at all. In Northern Europe, in Norway, we never really used much urso, but even less patients received urso after the Linder trial. I think that the current standing of urso is that if patients have an alkaline phosphatase response or a biochemical response, some evidence of a benefit, you give them a trial of, say, 3 months, 6 months, and if there is a response, then you keep them on. But you don't really know. And if you go to the next slide, this is an important point, because it impacts also on how to interpret the results of the many ongoing clinical trials in PSC. As you can see here, most of, if not all of the trials with ursodeoxycholic acid, they have a positive effect on the laboratory results of the patient. So apparently there is an effect. There is something going on. Alk phos is going down. However, despite all the research effort, we haven't really been able to prove that this translates into transplant-free survival benefits and overall benefits. But we keep prescribing urso if we see this laboratory response, and we feel that in those patients we're doing something useful. And in a very brief statement, you would say that this is exactly what is happening in all ongoing clinical trials. And if you move to the next slide, you can see that the presumption that ursodeoxycholic acid is working is based on alk phos going down. So you're looking at the same type of efficacy that we're currently seeing for urso, and you're saying this is something which is good. Ursodeoxycholic acid, it's a kind of a super ursodeoxycholic acid. It's slightly modified. And you can see that in the trial, the phase 2 trial, you can see that alk phos is going down, and it's going down in a dose-dependent manner. And really, they didn't have much problems with side effects. So this is one of the drugs that Gideon was referring to, which is taken now on into a phase 3 trial. Some of you may be participating in this, where you try to expand on this effect, looking also at other parameters beyond alkaline phosphatase. So is this more or less of an effect than what we already see with urso. We don't really know yet, but we hope so. If you go to the next slide. A similar situation is for this system, which is shown to the left. It's a bit of a complicated slide, but basically the orange cell at the bottom is in the gut, and the brown cell are the liver cells. And you can see that there is some signaling going on there between the gut and the liver. And one of those signaling molecules is called FXR. And you can see it in the gut and you can see it in the liver. And this inhibits bile acid synthesis. In PBC, there is now a licensed drug, Ocaliva, which works through this signaling pathway. It looks like FXR and it works like FXR, and it helps PBC patients. And it apparently also does something good for alkaline phosphatase in PSC patients. Then again, as the question I asked for ursodeoxycholic acid and for norursodeoxycholic acid, does this alk phos going down actually mean that patients will do better, that their strictures will improve, etc.? We don't know. And I think I share the skepticism of Gideon that this measure may not be a complete reflection of what's happening in the patients. Of course, a problem with this drug, as some of you may have heard, is itching. Many patients experience itching, and if you have itching already, it definitely gets worse. So I wouldn't say it's a dream drug for PSC, but I find it very interesting that you see this alkaline phosphatase response. And if you go to the next slide, you actually see somewhat a similar response with a completely different molecule, but it works in the same way. It also looks like FXR and it works like FXR. And you see this alk phos. So you have two different molecules giving you this alk phos signal in this system. And if you go to the third slide, next slide, you would see that there is another molecule in this gut cell at the bottom there called FGF19, which is kind of the messenger from the gut FXR signaling to the liver. And if you give a molecule which looks like this FGF19 guy, you also see a signal. Not really that much with alk phos for various reasons you could speculate. There is some improvement in liver biochemistry. And also interestingly, here there is also seems to be some signal coming from these fibrosis markers that Gideon was referring to. So clearly something is going on in this system. What that will ultimately mean for the patients, I don't know. There are also further drugs coming into play here because of the interest, not only in PSC, but also in PBC and also in fatty liver, where these drugs are being tested. If you go to the next slide. There are two big disappointments, I would say, and I will discuss a little bit later on what to make of them. The first one is the trial on Symtosium. Some of you may even have participated in this. We were very enthusiastic about this. It's an antifibrotic, so it should reduce fibrosis. It was tested not only in PSC, but in a variety of diseases with fibrosis. And as you can see, when these lines are basically overlapping, there is no effect. There is no difference between the placebo and the antifibrotic groups. And really, if you compared liver biopsies within these different groups, there was actually no change. So at least for this antifibrotic, it didn't really work. Whether it means that antifibrotics in general won't work, I'm not so sure. There are more antifibrotics coming now into clinical testing. Maybe we just did it wrong the first time, but certainly it's not super encouraging. Next slide. This is another, I would call it a big disappointment. This is a complicated slide, but the point is really that lymphocytes, immune cells, they can travel from the gut to the liver and make trouble. And this travel or this journey of the lymphocytes, you can block. You can block the molecules in the liver and in the gut that makes the lymphocytes able to enter into the tissues. And this is a drug which we're now using in our IBD patients. It's a very nice drug in many patients with a severe ulcerative colitis. And then the question is then also this a wonder drug for PSC? Are these immune cells coming from the gut really that much of a problem? Maybe they are, but blocking them with vedolizumab apparently does not really improve liver biochemistries. And this we don't know from any clinical trial yet. There was a big clinical trial in planning. I don't think it will ever happen. And the reason for that is that if you look at IBD patients who also have PSC, there is not really much of a biochemistry signal. There are other molecules into this machinery which are also under clinical testing. So I'm not saying that this does not mean that anything else won't work, but it was kind of a disappointment. If you go to the next slide. Antibiotics. Johannes already talked about this, so I will be fairly quick. The point is that if you look at the different trials of antibiotics, there seems to be something going on with ALK-Phos, like we saw for ursodeoxycholic acid, last week, like we saw for norursodeoxycholic acid, like we saw for the FXR FGF19 signaling pathways, as we didn't see for antifibrotics or vedolizumab. But for many antibiotics, there is something happening. And if you click now step by step, if you go first, click once. This is apparent with vancomycin. If you click more, with even other antibiotics, and click one more. Lifetime. I think you have to go back to the first slide and reflect upon this. Patients, antibiotics in a lifelong manner. I don't do that. I think, like Johannes said, we have to understand why this is happening. I think it tells us that the gut is important, that the gut microbiota is important. But before we start applying long-term therapies, we have to understand really what is going on there. And this is the research which we started out this session presenting. Next slide. Next slide. Then the point on immunosuppression. We all talk about immune system and PSC and PSC being an autoimmune disease. There have been many trials. I would say most of them not very good trials, but still they have been done, testing a variety of immunotherapy. And however you look at it, they don't really work. If they were to work, you would see this bottom diamond either to the left or to the right, but there is really no effect. And if you go one further slide, even in the context of transplanted PSC patients, we give a lot of immunosuppression. It is a little bit of a variable story. Some centers give a lot of immunosuppressions, others give less, but anyway, there is immunosuppression. And if you click one more slide, even in this more extreme setting, there is recurrence of PSC. We know that. So I think that the whole story on immunosuppression, it goes a little bit alongside the vedolizumab and antifibrotic story. We don't really see much of a signal there. And if you click to the next slide, I think this is then the summary. And we asked this question on the first slide, where do we go? And the question is, where would you go? I mean, if you were the doctor doing research in PSC, we have these bile acid drugs, urso, norurso, these signaling molecules, even more signaling molecules. You have seen the signals from antibiotics and fecal transplantation. And there is something going on there with ALK-Phos. We don't really know what that means yet. We're not really sure it translates into a transplant-free survival benefit, but there is something going on there. With immune modulation therapy, antifibrotics, there is really nothing going on. So if it's an obvious question. If I were to put my emphasis of research into one of these categories of drugs, I would certainly choose one of the two upper categories. And I think that's where you would see the first drugs, new drugs in PSC appearing on the market, being available for patients. I'm not saying that this bottom category won't work. Maybe we're just doing the wrong molecules. Maybe there is a very clever way of manipulating the immune system that we haven't figured out. And then we would also see drugs emerging there. But certainly I think that the next step is happening in these two upper categories. categories. And then if you click to the last slide, I think it's worth to remember this. And I think it's pretty clear that we're quite to the left in this journey of hepatitis, which you see here, when we're talking about PSC. We have drugs, they don't really work that well. There are still the problem of side effects, but I remain hopeful with time. I'm not saying how much time. I'm not saying it will take the same amount of time, but I'm sure that over the years, over the next 10, 20, 30 years, we will see better and better drugs. In 10 years, we will have some drugs. In 20 years, we will have even more drugs. And I'm also quite confident that we would be able to achieve, if not cure, then at least being able to block disease in PSC quite completely at the other end of that effort. So with that, I would like to end the talk and thank again everybody for attending and for the opportunity to talk.
Okay, thank you. If you both can turn on your video screens, and we will start our Q&A questions. So again, Dr. Hirschfield unfortunately had to go to clinic, so these questions are going to be directed to Johannes and Tom. So Johannes, first question for you. I hear these many testimonials about probiotics controlling IBD and PSC. What are your thoughts about the use of probiotics?
Oh, that's a great question. Probiotics is it's a hot and very interesting topic for many. There are many products available, and there are many things to say about this. But first thing is that probiotics is just not probiotics. There are a wide variety. There are different bacteria in them. I think there's widely variable. How many bacteria are they at all viable? Things like this. So there are many things to say about probiotics as a whole. When it comes to probiotics in disease, I think we know far too little. Many have been tried in many different conditions, but it's extremely variable which have been tried. Very often, very small studies, hard to conclude about anything. And then scientists will then try to pile everything together and say, oh, probiotics seem to work. While in practice, you have perhaps tested 20, 30 different things. 20, 30 different things. And to really pile everything together, I think is not the right thing to do. We have some very small studies of probiotics in PSC with no signs of effect. In IBD, you have some data suggesting that it could perhaps help keeping your IBD in remission when it's in remission. But overall, I would say we have very little evidence that probiotics, the ones that are available now, actually work. And then it's the question, should you then use them? And I think if you think of you like to use something that's good for your health, and then you try probiotics, I don't think there's any particular reason to do so. However, many people have some abdominal complaints and have tested probiotics and feel that it works somehow for their symptoms. And if it does, then I think it's probably okay. But you have no reason to believe that it really is good for your health overall and long term. But this is something we need to know more about. So I strongly encourage studies into probiotics. But I'm not sure the ones on the market now are the right ones. We have a lot of hopes also for maybe what we call next generation probiotics, which is based on better evidence on what's actually good for your microbiome based on the more recent data. So there are some new opportunities there, but none of them have been tested in PSC at least. So if that somehow answers the question.
Sorry, I think so. So Dr. Karlsen, if you could comment about the use of steroids and PSC, why do you think that steroids have been found to be ineffective? Do you think that the steroids are targeting the wrong cells, or have they just been initiated too late?
You're muted, Tom.
It's an interesting question. I mean, we keep talking about PSC as an autoimmune disease, and in autoimmune diseases, steroids should work. I mean, they don't work in PBC either, which is also an autoimmune disease, very surely so. So apparently there is something about these biliary diseases which make them rather resistant to immunosuppression. If you would ask my personal opinion, I think it's part of the evidence also that there is some driving factor to PSC. I'm not saying it's a bug, I'm not saying it's a virus, but to me, this lack of efficacy also tells me that there is something which is still there, which is driving the disease regardless of the immune cells that we're targeting with steroids. I don't think you have evidence to say that if you were just early on with immunosuppression, you wouldn't have PSC, because we see PSC happening also in IBD patients receiving immunosuppression, including steroids. So my personal opinion is I think it wouldn't work at any stage, even the early patients. I think that there is something going on with PSC and the disease development, which is just simply resistant to immunosuppression.
Thank you. Back to Dr. Hove, if you can comment a little bit more on fungi and how they kind of affect PSC, what their role is. And in particular, would you consider an unrelenting skin fungal disruption an indication of fungal disruption in the gut?
If you take the last question first, I will say that if you have a fungal infection in the skin, I think it's unlikely that has anything to do with what's going on in the gut when it comes to fungi. Or at least that will be a very extreme situation. So I think that's unlikely. When it comes to what's actually going on about fungi in the gut, I think this is an area of the microbiome field where we know very little actually. There are quite few. It's a bit more difficult to study them. So you really have to work hard to get the DNA out of your samples to be able to catch those few threads of DNA that are in there. So I think it's hard to speculate. What they say in this one paper about it is that it's, there tend to be some interactions between the fungi and the bacteria in the gut. So, and that these somehow are disturbed. But honestly, what this means, whether this has anything to do with cause, or rather is an effect of I would think there's a change in the gut environment when you have the cholestatic liver disease, is perhaps more likely. So this needs further studies. That's all I can say, unfortunately.
Tom, if you could comment on the study that came out last year regarding statins in PSC. And I guess for both of you, are you both routinely prescribing statins to your patients?
No, we are not.
Okay, neither are we, but
no, I think it's it's an interesting signal. I think statins are doing many good things in liver disease in general. We're also a bit afraid of it because sometimes we actually see toxic effects in the liver from statins. So it's a kind of a two-edged sword. If you were to take my guess onto that, my personal opinion would be that it's probably related to the end-stage patients where you have advanced liver disease, where actually statins have a beneficial effect, and that may be reflected in these data. I think there is also the possibility. I mean, we actually investigated statins in a study of the gut microbiome, actually testing what is the statins doing in the gut, because they're actually molecules which are derived from an old antibiotic. So they have actually some gut activity. Honestly, I think the study we did was not really big enough. We saw some effects, but if you were to be very speculative, maybe there is also some indirect effect going through the gut. But my personal bet would be that it shows some beneficial signal because of the beneficial effects on the liver disease, at the late stages, but not really on the bile duct disease. I wouldn't be using and reflecting upon statin as a drug working in bile duct disease.
Thank you. Dr. Hove, do you agree?
I fully agree with Tom. I have nothing to add, actually.
Okay. And so what do you think about changing our diet if you have PSC? Should we be switching to a plant-based diet? Will that change the microbiota for good? Or any other diet advice you could give?
Thank you for this question. This is a really difficult one. We know, unfortunately, nothing about the effect of diet in PSC. This is really a big black box, and worth an intense emphasis, because I think diet is one of the major factors that influence whatever is going on in our gastrointestinal tract. So with that to say, I could only recommend what would be publicly recommended dietary advice, really. So yes, American Heart Associated approved diet.
This is a big question for Dr. Karlsen. If you had unlimited resources, what would your ideal study be for PSC?
I mean, it's yeah, okay. I would try to find the causative agent. I strongly believe that there is some molecule, be that coming from the bugs in the gut, be that a protein, be that a smaller molecule. But I strongly believe that there is some molecular factor. And I would really try to race to find that. And we are doing that. We're nowhere near, but I would really try to find the cause, because I believe that that's the only place where you would really see cure. Anything else you would be doing would be decreasing disease activity. It would be making disease less severe, but it would progress ultimately, and it would always be there. So, yeah, that's would be the grand vision, and it might fail.
Okay.
Can I comment on that?
Yes.
I'm not sure you know if I would how important it is to go for the cause. If you can really find a strong modifying factor that you can treat, then you can live with it.
What about if someone is taking antibiotics for a different condition? How is that going to affect the microbiota? And then on the antibiotic line, can you comment on vancomycin? Does vancomycin kill T cells, or does it weaken the immune system in general?
I think, well, first question first. If you use antibiotics, it will change your microbiome. It usually reverts towards the normal a few weeks after you end your treatment. But it sometimes, not all bugs recover. So, and with repeated antibiotics, it's more likely that you have long-lasting changes in your microbiome, and perhaps lose important bugs forever. But this is hard to predict, and we know little about it. But in general, we have somehow, at least after just one round of antibiotics, it tends to revert back to whatever it was. When it comes to the effects of vancomycin, I'm not really a biggest expert on the effect on the immune system, how it may modify the immune cells, potentially in a beneficial way. I'm not always kind of has an immunomodifying or suppressive effect. I don't really know the details about that. What I know about vancomycin is that if you use it orally over time, you will really suppress a number of so-called gram-positive bacteria, many of which also are important. They are involved in bile acid metabolism, for instance, and you will have a really rich niche for many bacteria that I would often, in general, speculate could be less beneficial, like gram-negative bacteria, some of which cause infections. But I'm not sure whether we know if long-term vancomycin actually predisposes you to infections with these things, but that's what will be going on in the GI tract. So you will have a major change in your gut microbiome. That's been shown. Not in PSC patients, we have data, but from other conditions.
Dr. Karlsen, are you, if you have a patient with PSC that you're seeing for the first time, just wanting to know your approach, are you prescribing urso? And then on a second note, a participant wants to know why people that are on urso feel better, and when they stop the urso, their alk phos numbers get worse and they feel worse.
Yeah. No, I think that there are parts of those questions that you can answer and there are parts that you cannot answer. I mean, to the very practical point, I think that the trend now is what I said, and is what we are trying to do. I mean, actually, we're struggling a little bit with reimbursements because of the Linder trial and the lack of evidence. But you would, in patients, if you have an elevated alk phos, you would be doing a trial of ursodeoxycholic acid for, say, 3 to 6 months. If there is a significant drop, you would continue. Otherwise, you would just take it away. If you have no elevation of alk phos, I mean, I had a patient last week actually coming from another center in Norway with no elevation, normal biochemistry, but clear-cut PSC, IBD. And we had this discussion on urso. And of course, in that patient, I wouldn't prescribe ursodeoxycholic acid. I would keep it away. And if that patient then would be taking ursodeoxycholic acid and feeling better, I would not really know what to make out of it. But I think alk phos and the biochemical response is what you can use as a parameter of those patients in which you believe it may hold some benefit, and for those that possibly do not benefit. And exactly what is the difference between those two categories of patients, nobody knows. Yeah.
And what do you think about urso and decreasing risk of colon cancer in patients with?
I think this is an unsettled topic to me. I wouldn't be using it on that indication. No, we don't.
Neither do we. Dr. Hove, with all your research on microbiota, are we years from finding out really the answer to this, or is this ready for prime time in the short term?
This is, of course, an important question also. To say that we are ready for prime time, that would be a major exaggeration. I think we are where we are in general is that we have some findings that we find interesting, but we are not there that we really have identified what we think of as key factors. So we're not like planning a trial to modify that factor yet. So that means in terms of treatment, at least from our side, we first have to be there that we can decide on something that we want to manipulate, and then you could try to establish a time schedule. When it comes to more like biomarker aspects, that could come faster, but whether it's always a bit hard to make those clinically relevant. So that would also suggest that there are some years, unfortunately. That said, I think we try to design our studies and research now, really aiming for clinical translation. So our major headline these days is clinical microbiota medicine. So hopefully, if we say it, then we can believe, and it will happen.
And do you know how long after a drug is approved is it released? And the answer is going to be different in Europe than the United States.
Tom?
I mean, there are different regulatory paths in Europe and in the United States. I mean, FDA has its set of rules, and EMA, the European Medical Agency, has its set of rules. There are also national rules within the different European countries. So I think there is no preset answer to whether US would have it first or Europe would have it first. I think it depends a little bit on the drugs and the evidence. I think that the drug companies are pretty clever these days. They design the trials so that eventually, if they have a proper efficacy signal, it would be approved both in the US and in Europe, and we would just be talking maybe some weeks here and there with delays. So a good drug would come to both continents at the same time. That's my feeling. Yeah.
And what's the status on any pediatric trials?
That's a good question. I think that there are, I think you have been doing well in the US with your network. And when it comes to clinical trials, of course, there is all the emphasis on vancomycin. There is some emphasis on, immunosuppressants going in other directions because you have more of this inflammatory phenotype. I think in the end, if you have a drug working in the adult population, it should also work in the children with PSC. I believe it's the same disease, just in different phases. And I think what you would boil down to then in considering whether to prescribe it or not would be side effects, safety in children versus adults, etc. So even though there is not too much going on, I still think that there is a lot of crossover coming from the adult field whenever there is some breakthrough. Yeah.
And Dr. Hove, looking at the microbiome studies, is it more applicable, do you think, to IBD than PSC or both?
A good question also. I think it's very much applicable to IBD, in particular ulcerative colitis, which is more homogeneous than Crohn's disease. But I definitely also think that it's relevant for the liver disease. The thing is, actually, in our PSC patients, we find very little difference in their gut microbiome in the patients that have IBD compared with those without IBD. And what does this mean? We have been wondering a lot about this. I have a feeling that this is really driven by the liver disease. And if the status of the microbiome, the composition, its functions, if that's actually relevant for the disease progression, then it's similar in those with and without IBD, then I think it's really, we could manipulate it, and then we can also affect the liver disease. So I think it's relevant both for the intestinal disease and for the liver disease.
And I think I'm going to end this session with one question for both of you. How confident are you that there will be a cure for PSC?
Yeah, I don't know who wants to go first. I mean, I can go first. It depends on several things. First of all, it depends at what time. I mean, whether you're talking in 5 years, 10 years, 20 years. I personally would be optimistic that at some point we would be able to stop the disease. The problem is, of course, that many patients, also by the time we diagnose patients these days, they would already have scarring. And it's a little bit like in, when you have a joint destroyed, your hand destroyed in rheumatoid arthritis, at some point, I mean, whatever drug you give the patient, the joint is still not good. So you have to do surgery and you have to have the prosthesis. So I think to be able to stop the disease, I remain optimistic. That's otherwise, why would we be doing what we're doing? I'm optimistic we will be able to stop the disease. If we do it early enough, then definitely you could start thinking about mentioning cure, because maybe you would be at such an early stage that the liver actually can regrow. But at the late stage, I am afraid that in some patients, it would still, if you had the perfect drug or the perfect thing that you could take away, it would still be too late. Yeah.
Do you want to also answer, Dr. Hove?
I think Tom has very important points here. I think we could add to these that there are diseases of some similarity where treatment has, I would say, has significant impact on disease like PBC, where at least it seems like to have drugs that slows the disease down and reduces the number of patients that reach the endpoint that needs a new liver, etc. So I think based on that type of observations that we need to be optimists. If not, we would never be working on this.
Absolutely. Well, thank you both. I think this is a great segue to introduce Ricky again. And again, part of the research and advocacy is really because of all the patients in your group who are such a positive advocate for more research and for drug development. So we owe a lot of the future upcoming trials to you and your efforts. So thank you, Ricky.
You're welcome. So thank you so much, Drs. Hove, Hirschfield, Karlsen, and Forman, for your really wonderful updates today. I wish we could pick your brain even longer. I personally feel really hopeful and encouraged about the future, and I understand you're talking long term, but this is exciting. It's good to know as we continue to grapple with uncertainty during these unprecedented times that today's 4 medical experts haven't wavered from their goal of helping us discover effective treatments for PSC. So thank you all, it was fantastic. I have just a few reminders. On October 23rd, we presented Making Our Voices Heard PFDD Forum. It was a momentous day where over 40 PSC patients and caregiver speakers, plus our phone callers and comment writers, all presented their unique PSC journeys, discussing their symptoms, their impact on their quality of life, as well as talking about current and future treatments to inform the FDA, pharmaceuticals, PSC researchers about our community's many unmet needs. If you would like to add your voice, you have 2 options left. One, you may send in a comment before tomorrow's deadline by going to our website homepage and filling out the comment forms. In addition, soon we are going to be posting instructions for patients who would like to send us a video of their patient journey, so watch for that. If you missed the PFDD forum or you'd like to listen in again, please go to the link on our website at pscpartners.org. If you haven't yet joined our patient registry, please do so now and help advance PSC research. Sign up for our mailing list so that you can keep up to date on all of our programs. Visit links to updated COVID-19 information. Watch for continuing Zoom room events for both PSC patients and caregivers. Feel free to send us your comments about today's webinar at contactus@pscpartners.org. Thank you again to our outstanding speakers and our webinar series sponsors, our conference co-hosts, University of Colorado Health and Children's Hospital Colorado, and our other corporate sponsors, Gilead, Mirum, Intercept, Albireo, Shire, and High Tide for your support. Please stay in touch and thank you for joining us today.